Hi, I will be doing my part of the 6.3 presentation, which is the last two orange boxes of the textbook talking about HIV and Florian chain. So I will start by talking about HIV. HIV is a retrovirus which has genes of RNA, but then they use reverse transcriptase to make DNA in order to make copies of its genes to make another virus. To make a copy of itself. And yeah, basically all of this happens when they enter a host cell. So before I explain anything else, our immune system produces antibodies from cells called the T helper cells. So the T helper cells signals and makes antibodies and signals them to fight off any bacterial infections of the body, like any foreign substances of the body. So in this case, HIV is a retrovirus. So once it comes into a host cell, our immune system will immediately call the T helper cell, which will make and produce antibodies that will fight off HIV. But the thing is with HIV invades T helper cell and destroys them. So by destroying T helper cells, it will take a toll on the production of antibodies. So without T helper cells, they can't make and signal the antibodies to fight off the retrovirus. The rate at which the T helper cells are destroyed by HIV varies, but it can be slowed down by using antiretroviral drugs. So in most HIV positive patients, antibody production eventually becomes so ineffective that a group of opportunistic infections, this guy right here, strike, which would be easily fought by a healthy immune system. But since the immune system is not healthy anymore, they can't really fight off the infections, which of course these guys will make a strike. So several infections are so rare that they are marker diseases for the latter stages of HIV infection. An example of one of those diseases is Kaposi sarcoma. Kaposi sarcoma is a cancer that causes patches of abnormal tissue to grow under the skin, in the lining of the mouth, nose, throat, and in lymph nodes or in other organs. So having several diseases or condition is called a syndrome. And when that syndrome is due to HIV, being present in the patient is said to have AIDS. AIDS stands for Acquired Immunodeficiency Syndrome. The virus only survives outside the body for a short time, and infection normally only occurs if there is blood-to-blood contact between infected and non-infected people. There are also various ways in which this can occur. One is sexual intercourse. Another way is transfusion of infected blood or blood products such as factor VIII. Another way that you can get AIDS is by sharing hypodermic needles of by intravenous drug users. So to cap what I just said about HIV, HIV is a retrovirus that has genes made of RNA, but once they use reverse transcriptase, they make DNA to make copies of its genes once it has entered a host cell. Our immune system produces antibodies by a complex process, and includes using different types of lymphocytes, including T helper cells. So our immune system calls our T helper cells to make and signal antibodies to fight off any bacterial infections or foreign substances that enter our body. But what HIV does is they invade and destroy T helper cells, which can no longer produce antibodies to fight the foreign substance. However, that process can be slowed down by using antiretroviral drugs, but some don't really work that way. So eventually antibody productions become so ineffective that a group of opportunistic infections strike, which would be regularly fought by a healthy immune system. But since we don't have that, they strike. Several infections are so rare that they are marker diseases for the latter stages of HIV infections. So one of the diseases that you can catch with for the latter stages of HIV infection is Kaposi sarcoma, which is cancer that causes patches of abnormal tissue to grow under the skin, in the lining of the mouth, nose, and throat, in lymph nodes, or in other organs. So having several diseases or conditions like that is called a syndrome, and if that syndrome is due to HIV being present in the body, the patient is said to have AIDS. AIDS is acquired immunodeficiency syndrome. AIDS spread by HIV infection, of course, and the virus only survives outside the body for a short time. An infection normally only occurs if there is a blood-to-blood contact with a infected person and a non-infected person. Other ways of catching AIDS or getting AIDS is through sexual intercourse, transfusion of infected blood or blood products such as factor VIII, or sharing hypodermic needles by intravenous drug users. Moving on to the next orange box, which is about Florey and Chain. So in the late 1930s, Howard Florey and Ernst Chain formed a research team in Oxford. They investigated the use of chemical substances to control bacterial infections. So the most promising research they've had was discovered by Alexander Fleming in 1928, which was penicillin. So by growing the fungus Penicillium in liquid culture conditions, it simulated it to secrete penicillin, which killed bacteria on agar plates. Since it killed bacteria, they wanted to test whether or not it would control bacterial infections. So, experiment time. So they started testing it on mice. They using 8 mice as their test subjects. They split it into 2 groups. One group of 4 mice was injected with penicillin, while the other group was not injected with penicillin. And 24 hours later, the mice that were injected with penicillin are healthy, while the ones without were dead. This experiment concluded that penicillin worked against serious bacterial infections. So now they wanted to test it on humans. The first patient that they tested it to was a 43-year-old policeman with an acute life-threatening bacterial infection that was caused by a thorn scratch. So after giving the guy penicillin for 4 days, his condition improved, and when the supply ran out, he died. He relapsed and died. After that, they produced larger quantities of penicillin and tested it on 5 more patients with acute infections. After giving them the penicillin medicine, 4 of them lived, but the other one died. Only one of them died, and the one who died was a child. So basically, penicillin weakened his artery wall, the artery that carried blood to his brain, and even though he was cured from the infection, the child died suddenly because of brain hemorrhage when the artery busted. So now pharmacies in the US then began to produce penicillin in much larger quantities, allowing more extensive testing, which confirmed that it was highly effective treatment for any previous incurable bacterial infections. So to summarize what I just said, Florey and Chain, a dynamic duo that created a research team in Oxford in the late 1930s. Alexander Fleming in 1928 discovered penicillin by growing penicillium, which is a fungus, in liquid culture conditions, which secreted penicillin, and that penicillin kills bacteria on agar plates. Testing it on mice first, they split 8 mice into 2 groups. 4 of them were injected with, and 4 of them were injected without. 24 hours later, the one that was injected with penicillin lived, while the other one died. After testing it on mice, they concluded that that penicillin did control bacterial infections. So now they wanted to try it on humans, and their first patient was this 43-year-old policeman with an acute and life-threatening bacterial infection that was caused by a thorn scratch. After giving the guy penicillin for 4 days, he was like, oh yeah, okay, my condition improved. But then after his supply ran out, he died dead. So after producing much larger quantities of penicillin, they tested it again on 5 more individuals. After couple of days, they found out that 4 of them lived, while 1 died. And the one who died was a child, because penicillin weakened his artery wall that carried blood to his brain, which, when was the artery busted, it resulted to brain hemorrhage. So he didn't die of his acute bacterial infection, but because of penicillin weakening his artery wall, which busted, he died of brain hemorrhage instead. So now penicillin is being sold in larger quantities of medicines from pharmacies in the US, which confirms the effective treatment for many of the previous incurable bacterial infection. And that is it, what you need to know about Flori and chain.